So welcome back. Last time we saw the idea of seed dispersal, getting seeds around, and this time we'll look at what happens to seeds after they get into a good place for growing. So illustrated here, this is a whole bunch of coconuts. So coconuts, of course, are seeds, well adapted for actually floating around on the ocean. And should they land on a new shore, they have a supply of fresh water in there and some nutrients to get them going, and pretty well adapted to growing on beaches somewhere. So this is kind of the fate of a natural coconut. So what's inside of a seed? Just a reminder again, here we have seeds, basic components found in all seeds, or I guess generalized seeds of all seed plant groups. We've got the seed coat around the outside, so one level of defense potentially against the outside world. That defense against the outside also could be achieved by some component of the fruit in the fruiting plants, or part of the cone in um, the non-flowering seed plants. Also in there, very lovely embryo here. We'll go into a little more about the structure of the embryo, but in this micrograph here, nicely distinct from the rest of the insides of the seed here. And then basically, apart from the coat and the embryo, this is a seed filled with food for the embryo. We saw last time that there are different potential foods for the embryo, so not always present, but when they are present, that could be either the endosperm, which we saw in the flowering seed plants, or potentially that would be gametophyte tissue if we're looking at the non-flowering seed plants. So inside of there, an embryo, and then the process of germination really gets this embryo out and growing on its own, starts growing a root structure, starts growing a shoot structure, and that's the beginning of the whole new plant as those organs and regions of the plant get established and start providing for themselves. So I think it's kind of interesting, I don't know, a little cultural tie-in here. There's a big push sometimes for foods that have whole grains, right? I think my favorite one these days is popcorn. Popcorn is whole grains. What does it mean to be whole grains? Well, whole grains is basically having all parts of the seed. The grains are the fruits, technically, of the grass family. So lots of grains that we eat, some that you can see on here. So wheat, oats, barley, rye, millet, corn is in there. I'm sure I missed one, but lots of similar foods that we get. We like them for their starchy components, but there's more than just starch. So if you take one of these grains, technically it's a fruit. So it's a special kind of fruit where the fruit wall is the outside and it's actually fused to the seed inside. So it's one seed inside of a fruit and actually fused to the fruit wall. The fruit wall can be kind of tough. So when you get your rolled oats, this is a process of basically grinding those up, flattening them out so that the starchy insides can be accessed and you don't have to, say, cook your way through the outside. Sometimes you get oats that look a little more like this. You gotta cook them for longer. But a lot of times our grains are also ground up to make use of them. So in the grinding, if you take the purely ground up bits, you get ground up bits of the starchy endosperm, ground up bits of the seed and fruit wall, which we call the bran here in this grain anatomy. You get ground up bits of the germ. Maybe you've heard about wheat germ. In the more highly processed versions, you would get parts that are purely endosperm. You could buy wheat germ separately. So if you get white flour is a little more purified in for this endosperm portion. If you get wheat flour has a little bit more of the bran left over in there. So you can enrich for bran and really seek that out. We saw the dietary benefits of fiber So bran is largely not digestible. That has the benefit of fiber in your diet. The endosperm is the more nutritious, starchy portion here. And whole grain, of course, includes all of those. So I think it sounds nice. Really, you're getting uh, some fiber with the bran if you get whole grain as opposed to purified endosperm. And then also the germ, because this is the portion that would turn into the plant, this would have a little more protein. Um, I don't know how much more protein, but a little bit more maybe than the starchy storage product here, because those are cells that are going on to make up the new plant if this thing were ever to germinate. All right, so how do we get from fertilization to embryo? Zygote is the first cell resulting from fertilization and basically takes on this kind of elongated and then eventually this heart shape appearance. And that's even how they call it. This is the heart stage of embryo development. This is in a dicot, so a plant that has two cotyledons. So up here, this heart shape is because two parts of this are going on to become these cotyledons or the seed leaves. And it's kind of setting the stage for the seedling to be made. Maybe a little bit more detailed about this. This is an embryo that's colored to show the fates of the different cells. So you can still basically count the individual cells that make up this embryo, but they're shaded with what they're going to become in the seedling. So parts of this are going to become these cotyledons. Parts of this, the red portion there is very important as the shoot apical meristem. Purple portion here is the root apical meristem. Part of this set out to be the root, part of it as the, what we call the hypocotyl. So kind of an interesting seed, embryo and seedling feature is this region in between the root proper and the shoot proper. It's called hypocotyl. And sometimes that has a very important job in, say, like digging into the soil to establish the seedling. Basically, we've got all the parts ready to go. So meristem, parts that are root, parts that are shoot, and that's what they'll turn into. And this is from studies in model organisms, probably Arabidopsis for the most part. And you can actually trace the fates of individual cells as these things are developing. So I mentioned cotyledons in the previous lecture, and then also a little bit today already. I like to call them the seed leaves. I think cotyledons sometimes can get a little bit of a mouthful there. So seeds have them as part of the embryo, and we have in the flowering plants I mentioned, we have these monocots and dicots. The favored term for most of our dicots now is eudicots. You can ask me about that one if you want to. So embryos that have two cotyledons shown by the arrows here, so rather long in this plant in the mustard family. There are two of those. So this would be the portion, this would be the apical meristem ready to go, two seed leaves, cotyledons, and then down here would be the root portion ready to go, and then the seed coat on the outside of that. So two here, a little bit harder to tell in the monocots, but there is technically one of these seed leaves, one cotyledon. So if you germinate these, if you grow corn, first thing that comes out of your corn plot is a single leaf, right? One cotyledon. When you plant mustard or squash or radishes or tomatoes or peppers or eggplants or most of our beets, most of our plants that we get in the garden are probably dicots, and you'll see those come out as two seed leaves when the seeds germinate. Conifers, they don't have to play by those rules, so they sometimes have many cotyledons here. And then just an illustration here, this is another eudicot in the bean group. And I mentioned last time when we talked about nuts, sometimes the nuts that we eat have their food, the parts that we like to eat, stored up energy in their cotyledons. So that's also the case in the bean family. If you split a pea apart, those two split halves of a pea or peanuts or beans, those are actually cotyledons, and they're connected together to the rest of the embryo. But if you just took the seed apart, the cotyledons would be most of the space inside of there. So sometimes the cotyledons have a function to store energy, sort of take up whatever was available energy as part of the seed, and they're store it as part of the embryo. So then the seedling doesn't have to get its food from the endosperm. The seedling already has its food basically stored up in the cotyledons. That's one function of the cotyledons. Another one is that they're basically pre-made leaves. So when germination happens, they can just jump right out of the seed and be ready to absorb some of the light and get photosynthesis going. Really, as soon as this plant can get enough light, then it can grow. It's making its own energy. It builds new organs, new leaves that get it new light and new energy. And a lot of plants can become self-sufficient that way very quickly. So a few functions related to the old cotyledons there. We talked about getting the seeds around and introduced the idea of trade-offs a little bit. So I talked about flower size and number as trade-offs. You can also look at trade-offs relating to the seed dispersal part of the life cycle. And so some comparisons here. We've got larger seeds, smaller seeds. Larger seeds are energetically expensive to make. So for the same amount of energy, you'd make fewer large seeds or lots of small seeds. This relates to something you've probably covered in ecology, right? Do you have a few offspring and invest lots of energy in them? Or do you have lots of offspring and invest relatively little energy in each of them? Both of these are suitable alternatives. They're both successful in their own ways. The plants that have them have probably found whichever strategy they have to be useful. So this walnut tree is investing in very large seeds, and each of those would have a lot of nutrients for the seedling that results. This St. John's wort here, lots of small seeds, each seed pretty low chance or low nutrient investment, but maybe across a lot of seeds, at least some of those will have a good chance of getting established. Another trade-off would be maybe the comparison in investing in animals or using something more abiotic to disperse the seeds. So lots of comparisons I could have made. I just chose dandelion here compared with a cranberry. So cranberry is a relatively large fruit, almost bizarrely large compared to the plant that makes it. So the plant that makes cranberry is this spindly little thing with relatively small leaves. It makes this giant cranberry. And so that's a lot of investment for this plant to put into that cranberry. That is an animal attractant, an animal reward, and even away from where humans are cultivating cranberries, they're still quite large like this relative to the plant. So the extra investment in the color, the nutrients, the attraction there, with the cranberry versus individual dandelion seeds would be two different strategies for getting seeds around. So getting animals to deliver your seeds somewhere useful maybe, or just relying on the wind to blow them around. This is something I like, I talk about trade-offs, and this is just a study that illustrates it pretty nicely. The size of the seeds and number of seeds per plant. So again, pretty clearly illustrating that plants in an area have basically similar abilities to get energy. And so what they do with that energy, that comes down to their strategy. So are they going to go in for lots of seeds or few seeds? And if they have the same amount of energy to give, if they're making lots of seeds, each seed gets less. If they're making few seeds, each seed gets more. And pretty nice relationship here, illustrating that pretty tight correlation of number of seeds and size of each seed. Another thing to consider when we think about seeds being output, reproduction of a plant. What does it take to reproduce? What does it take to keep a population going? So uh, something also comes up in animals. If you think about reproductive output in the animal, in the life of an organism, what does it take to increase the size of your population? What does it take to keep you stable? What would be so disastrous that it might cause your population to decrease in size? So basically, over the course of an organism's lifetime, it needs to replace itself, right? So of all the seeds that are made and all the seeds that go out in the world, if those seeds are at least successful enough to replace the organisms that made them, then the population will have steady numbers. So as individuals die, they're replaced by surviving individuals that can carry on the population. Populations grow if they introduce more individuals than the existing size of the population. The populations decrease in size if they are making fewer offspring than there are individuals in the population. So there's a pretty wide variety of investments in number of seeds. So we have a goldenrod here, one plant producing thousands of seeds, all going off on the wind, maybe not all having great success. Trillium here, this is a fruit capsule here. So not quite as many seeds, but maybe better chance individually. Uh, Hard to say. Other people study this. You could find lots of papers on these kinds of life history kinds of studies. But maybe just kind of break it down. If we look at an individual, right? So reproductive output. That's one of my worst drawn plants. Get this down there a little bit. Okay, so how many seeds per plant is dependent on, where do you find the seeds? So you got a number of well, on that same plant, you can have lots of flowers, a small number of flowers. So number of flowers or fruits, number of seeds per fruit. Multiply these together, that's your reproductive output per, let's say, per season. What about in a lifetime of a plant? So plants have, we have plants that are annual, and reproducing in one season is all they ever get. We have plants that are perennial, and they will reproduce every year for as long as they are able. So here's an example of a kid exploding a cattail and just thousands, I don't know, millions of seeds, so many seeds coming out from one cattail head going out into the world. So if we, I guess, add on to this, we're just sort of multiplying everything out here. So one plant, so population output is number of plants times number of flowers per plant. Put it on there, per plant. Number of seeds per fruit. And I don't know, maybe in a different year, number of productive bouts or episodes per lifetime. So however long that cattail lives, if it's the same plant alive for 20 years and it makes 4,000 seeds every year. That's pretty substantial output from one plant. And so there's also variety, there's just variety all over the place. Variety in the number of times an individual plant reproduces. So if we have a perennial plant, even a perennial herbaceous plant like our Jack in the Pulpit here, this plant is producing 20 to 50 fruits in a year. That was one successful year, but at the end of the year, this plant is going to go dormant, stored energy at the base of the stem here, and it's able to come back and flower and produce this many fruits again the next year for, I don't know, maybe indefinitely. Kind of an extreme example here. This is an extremely large palm tree. Takes years to grow this big, and once it gets to reproductive height, reproductive size, it produces this amazing floral display, millions of flowers, millions of seeds produced, and then it dies. The whole thing dies, just done. Used up all of its energy, destroyed its apical meristem in the process, and this whole ginormous tree done for. So lots of reproductive output once, but limited to one reproductive episode in a lifetime. So sometimes the seed production of 1 year, you look at the example of our giant palm tree over here, that reproductive output can be enormous in 1 year and maybe decreased or non-existent in another year. And the uncertainty of growing as a plant, and also kind of combined with the ability for seeds to stay dormant, this is something we don't see a lot of parallels in animals. There's some animals with eggs that can stay dormant for a long time. But a lot of animals, really, they've got to stay alive as adults every year. And they just don't have eggs that last that long or any kind of dormant period that lasts that long. So in plants, they have this phenomenon called the seed bank, where one plant, I don't know, some interesting fruits, too interesting. You'd think I could spontaneously draw a plant a little better than this. So, flowering, fruiting plant, in a year when it's producing seeds, let's just say those seeds end up in the soil. As we've seen with germination, they don't always germinate. And that's not always the worst thing. So if a seed doesn't germinate, it can stay dormant for a while. So give it a few fates for a seed in the seed bank. Seeds can stay dormant for multiple years, not uncommon. So a deposit into the seed bank is production of seeds. And let's say a withdrawal from the seed bank would be seed germination. And it kind of helps out plants not to have all these seeds germinate in a given year. That, let's say we have a bad year. And say all these seeds that germinated, say they fail to survive. You can kind of break the loop here, right? If you had all the seeds, if you exhausted all your seeds, caused them to turn into germinated plants, and those germinated plants died before they could produce seeds, that would be the end of your population. But fortunately, the seed bank stays intact. And so from 1 year to the next year, those same seeds that were produced in the year before, 2 years before, 5 years before, they're still there and they can try again the next year. So it's very much this term seed bank it grows on me more every year. It's very much like a bank account, right? So if you have extra money, you stick it in the bank. Literally, I guess if you're a plant, save for a rainy day, save for a good season. So you're not sure as a plant if this is going to be a good year or a bad year. Could be a drought year, could be a fire year, could be a crazy herbivory year, could be a disease year. And the seeds that germinate, they've got to commit to living as mature plants. But the seeds that stay in the seed bank, they are well protected, protected against the elements. They might be protected against fire, against drought, against herbivores, diseases. And so they're still there, even if things don't go well for the germinated seeds. So in good years, seeds are deposited into the seed bank. In, say, following a bad year, that might be a year of extra withdrawals from the seed bank. So say we've got 3 years of drought, uh, or I guess years with good wet springs, but say there's a disease running around, those would be seeds that would not survive. That's withdrawal from the seed bank. But eventually, some of those seeds are going to come back and germinate in a good year when the seed bank can be replenished again. So kind of a whole other layer on this question of replacing your population with seed production is that you'll have good years and bad years, and this reproductive output is not only covering for individuals that exist as adults now, mature plants, but it's also covering for future years, covering for episodes of complete destruction, planning far ahead. All right. So seed bank, as I say, influenced by these features here. So the more abundant a species is, the more seeds it can put in. Seed banks are influenced by where the seeds come from. So if you dig up a bit of soil, it will have the potential to germinate into a variety of seedlings, maybe reflecting the plants that are there already, maybe reflecting plants that have blown their seeds in from somewhere else, maybe plants that are no longer there as mature plants, but whose seeds are still in the ground after some time. So it's a pretty dynamic process, putting seeds in the ground and drawing those seeds back out through the process of germination. And I don't know, another one to think about that may not have the same parallels in animals. Seeds, if they are taking 10 years in between being produced and germinating, you can have seeds that are mixing genetic material, mixing genetic adaptations from different seasons of different years. So if you have a plant that's adapted to a hot year and did very successfully in the hot year, maybe it's got genes that are well adapted for that. Maybe if you've got another plant, different year, well adapted to colder conditions, and those were well suited for that particular year. It's possible that the offspring of those different adaptations will have a chance to mate together through this seed banking kind of generational skipping of time. Just, I guess, increases the, there's a little thought experiment here, increases the genetic variation that might be contributing to the next population that comes up from seeds. So they're not all coming from the same kind of adaptations. There might be wider adaptations, genotypes that are not always useful that sometimes show up again because they were useful once, and maybe they'll be again. So I'll get rid of that image for you. Transgenerational mating, why not? So seed dormancy, you can kind of just conceptually see the value of seeds being dormant if not every year is a good growing year, right? So some years it's just good for seeds to stay in the ground, at least some of them, because they will survive if the year is bad and it was a bad year to germinate, they'll still be there in the next year. This was an interesting connection that I hadn't thought about. I thought was pretty interesting as an experiment. Seeds that are dormant also, in this case anyway, can escape detection by seed-eating animals. So this is an example of the black locust, one of our trees that we can find around here. And here we have dormant seeds and germinating seeds. And the seeds taken, by and large, the seeds that were grabbed by the rodent in this experiment were the ones that were basically ready to germinate. So in the process of getting rid of the seed coat, for this animal, they actually became kind of smelly. They were easily detected, or more easily detected, as a food item. So seeds are alive, and during this process of germinating, they're breathing and interacting with the atmosphere and the environment. They are contributing some scent chemicals and allowing themselves to be found. So dormancy, for intention or maybe just a coincidental benefit, staying dormant also for these seeds allows them to escape detection a little bit more. So a good reason to stay dormant if you can. Certainly you don't want animals eating all your seeds in the off years either. Okay, so we had a lab exercise on germination. Germination is the process of the seedling emerging from the seed. It's a one-way street. So the seed is very nicely sealed up. As we'll see, there's some seeds that can do that for many years. But once the seed is open, once the seedling begins to emerge, that plant is then as vulnerable as any other plant. It can't crawl back in the seed and give up and say, no, I didn't want to. You can't go back to bed as a plant. So these seeds have to really make that decision very, very judiciously, one time, the right time to come out of the seed, because that's their one chance. So I guess conversely, there are signals that need to line up well to make the seed germinate under good conditions. So again, seed germination, start with seeds and something comes out. Very often, I think almost always, it's an element of the root that really makes it out first. So the root comes out first, gets a foothold, gets the seedling tied in with the water and soil nutrient sources that it needs. Maybe even a mycorrhizal association. So I guess if you want to watch this, there's individual seeds germinating there. The white part comes out first, that's part of the root. And then later the green parts come out, and some of these you can see the seed leaves on there. So anchor first, light seeking second for the most part. So germination is really just anything emerging from the seed coat, breaking that nice boundary between inside and outside. So what causes that to happen? We've done some experiments on that, and it's not universal. So the most universal signal is water. The plants you put in the ground, if you're planting a garden at home, these are plants that people have selected for germinating reliably. So when you get a packet of seeds, quite often they say, 95% germination. So you put 10 seeds in the ground and you were pretty well guaranteed to get 9 or 10 seedlings coming out from that. That's not always the case in wild native plants. That's something that people like because we have control over this whole process. We say, we put you in the ground, you're going to grow. So water important. Think about what else might be important. So light, lack of light. Some seeds germinate in darkness, but they won't in the light. Temperature can be a signal. pH, damage to the seed coat, as we've seen. So some number of features line up and cause this to happen. This is what you might see as a result of a germination trial. So this is just a cumulative percent of germinated seeds over time. So this should be a chart that only increases, so it's cumulative. And what it shows is just under a couple different conditions, the total number of seeds that have germinated is higher under this condition, a little bit less high over these other conditions. And measuring, here we go, light intensity. So here's a case where germination rate is pretty high at the low lights, and then at high light intensity, hardly any germination. So nicely illustrated there. Plant that would really prefer not to germinate in the light. So as you look at these signals, I think a lot of times it makes pretty good sense. So where does a seed need to be to establish? Depends on the species, but being away from the light would indicate that a seed has found a place, say, deeper in the ground, as opposed to lying on the surface of the soil. That's a pretty good cue for a lot of plants. Being lower in the ground allows them a better anchor to get started. Some plants actually prefer to germinate in the light. Those would be ones that are maybe easier, better able to get their root system established starting at the ground level. So just a study here showing the impacts of light as a germination signal. We have some wild type and mutants here. So these wild type is this call 0, and nobody's germinating here in this light regime. This is the far red, or the mostly dark days. No germination here, no germination here. And then these are mutants that aren't getting the signal just right. So this is a study illustrating the genes responsible for making that germination cue interpreted by the plants. And then another example here where the wild type germinates just fine. This is one where the darkness is interrupted by a burst of red light or bright light. And that's that signal to get growing. So big difference between the top and the bottom and the wild type, as opposed to whether, contrasting whether they've been in a long dark night or a short night. So day length, sorry, not just light, but also day length, I should say. So long days, short days, that can influence when seeds germinate. Scarification, we did this in the lab where you can scratch the seed coat. Seed coat is a nice preventive, a barrier to prevent the, as we saw, the herbivory by animals. Also just can be a nice way to ensure that the seeds that get in the ground, they don't all germinate right away. And the ones that do germinate would be the ones that have been kind of beat up by the environment for longer. So time, being crushed into the soil, kind of ground in there through natural weathering events, or maybe trampling. Scratch the seed coat enough so that it kind of gets a break where it starts getting some signals from the environment, water, air, something like that. And that can start germination. And highlighting here just a big difference in the top one here, not scarified versus scarified, much higher germination in the scarified seeds. And then this one, another one, these are water plants, and seeing the effect of passing them through the gut of animals. So a couple different kinds of fish. What does the germination look like after being eaten by these various animals? So this was kind of investigating how effective fish might be at transporting seeds inside their gut and still allowing those seeds to germinate. So scarification, that's another way to be scarified, is to basically get beaten up by chewing, digestive enzymes, things like that. We also saw cold stratification, so exposure to cold temperatures. And here we have different lengths of time. So seeds that are barely frozen, they don't germinate very well, but ones that have been stuck in cold temperatures for 6 months, those germinate pretty well. And this is pretty common across this group of plants here. So a little bit of time in the cold wasn't enough, but longer periods of time in the cold would cause these to germinate. A little more extreme, there's some plants that have adaptations that allow them to respond to the presence of fire or smoke. So a few examples here. These are conifers, a couple of these, and this one's actually a flowering plant from Australia. And you can see the top two, these are all singed up, and they have opened up in the presence of the heat from a fire. So when these cones are made on the jack pine, they're sealed up pretty tightly, and they've got a tight resin barrier in there, and that melts away under extreme heat and opens these up. Same thing for the Banksia here and the lodgepole pine. So these are plants that are adapted to disperse their seeds when they've been through a fire. Going through a fire, take a minute. Why would that be adaptive? Well, when fire comes through, a whole bunch of new nutrients are available. The burned bits of plants have released their nutrients back into the soil, and there's really low competition, right? If you can be one of the first seedlings to establish after a fire, there might not be a lot of other plants that survived that. So other seeds might not be adapted to return after fire, and if you are, it's a good place to be. This one I found was interesting. They actually used just smoke extract. So the chemical signature of fire, not just the heat of fire, but just the smell of fire was enough to get these seeds to germinate better. So plants are pretty crafty there. How long can plants live in seed banks? Kind of an open question, but there's some competition for the record holder here. This might be the oldest verifiable seed germination that I know about. This is a sacred lotus. So this is a plant that produces seeds that are very durable, and it's a water plant. So what they found were seeds deep in the sediments of water bodies. And this one traces, as you can see there, 1,200-year-old, give or take, sacred lotus plant. So over 1,000 years stuck in the mud, germinated, and produced the plant. So I'm sure there are older seeds, but this is one of the better documented examples there, and maybe less disputed. Here's another one. So kind of taking this ability of seeds to be dormant for a while and really putting that natural seed bank adaptation to human use. This is a project, an international project to store seeds of important plants. So a lot of economically important plants, basically storing up their seeds in case of a disastrous event. So say if there was a drought in a region or wildfire, or as the video is going to mention, political turmoil might cause political unrest, might cause farming to be put on hold for a while in a region, and maybe cause the loss of the seeds in that area. It's possible to store these away in this location way north up in the Arctic Circle in Norway, and they're storing seeds from all around the world in the event that people need them. So in these days of climate uncertainty, in a lot of regions, political uncertainty, this is a way for holding on to this like agricultural heritage that people have. Skip some video and go.
Pole. The Norwegian sovereign islands are the northernmost settlement in the world, home to just 2,000 people. And aside from a small number of researchers and tourists hoping to spot a polar bear, few people know of this place. Yet Svalbard actually plays a pivotal role in ensuring humanity's continual survival. So why exactly is this place so important? It's because of this. The Svalbard Global Seed Vault, sometimes referred to as the Doomsday Vault. Set 120 meters inside a mountain, this secure underground facility is capable of storing up to 2.25 billion seeds, collected from 4.5 million crop varieties around the world. Basically, what lies behind these metal doors is the ultimate insurance policy for the entire planet's food supply.
I always get this feeling of going into a cathedral when I get there. It's just so amazingly quiet, and you get this divine feeling. It's a history of agriculture in there, and it's potentially also the future of agriculture in there. So, to me, a very, very special place.
That was Marie Hager, Executive Director of the Global Crop Diversity Trust, the organization that manages the vault. Since it opened in 2008, the Trust has collected, catalogued, and stored 860,000 seed samples deposited by gene banks from across the planet. Those are huge repositories that preserve genetic material.
Russian seeds and Ukrainian seeds are literally on the same shelf. Yes, there are seeds from South Korea, from North Korea, well, basically from all over the world.
The seeds sit in 3 cold storage rooms kept at a constant minus 18 degrees Celsius. The cool temperature ensures that the seeds remain viable for decades, and in some cases, thousands of years, just in case the gene banks who donated them ever need them again. And ultimately, that is one of the major reasons why the vault was set up in the first place. While there are around 1,700 gene banks in the world, these facilities can be vulnerable to things like political instability, natural disasters, and war. And if the seeds inside these banks become compromised, the loss can pose a significant threat to global food security.
Ideally, we want to have a copy of each accession in every gene bank in the world in Svalbard, because we can hope that things don't go wrong in the world, but occasionally things do go wrong. And we have lost complete gene banks. We have lost gene banks in Iraq. We have lost gene banks in Afghanistan. The gene bank in the Philippines was first struck by a tsunami, and then it caught fire. So it's just illustrations of why it is important to have a backup system.
Until recently, no one had ever put this system to the test, but that all changed with the outbreak of war in Syria.
So one of the most important gene banks in the world is in Aleppo in Syria. Due to the war situation, the gene bank cannot operate as it should. The seeds are safe, but the land around the gene bank cannot be used, so they can't duplicate seeds, they can't share seeds with farmers and breeders and scientists.
The gene bank in Aleppo held ancient varieties of wheat and durum that dated back to the dawn of agriculture, some 10,000 years ago. Essentially, hundreds of crop varieties that had taken thousands of years to cultivate were at stake. Losing these crops would have proved devastating for global food security. So in September 2015, the Global Seed Vault received an urgent request from Aleppo, asking them to make a withdrawal.
And we have then been working with the gene bank in Aleppo for quite a few years now, and almost all of their collections are copied in Svalbard. They decided to take seeds out of the vault in Svalbard and then re-establish the gene bank. So you will see in the vault that there are some holes in the racks. There should have been some boxes there. So the seeds quite simply have been shipped back or have been shipped back to Morocco and Lebanon.
A total of 128 crates of seeds holding around 30,000 samples of wheat, barley, lentil, and chickpea, among others, were removed. But the seeds couldn't be sent back to war-torn Syria. Instead, the samples were shipped to Morocco and Lebanon until they were able to re-establish the gene bank in Syria.
They're being grown out there now. So they will re-establish the gene banks there, and when they have the first yields, they then will send seeds again back to Svalbard.
While the seed bank in Svalbard isn't the only solution to global food security, it is reassuring to know that seed varieties from all parts of the globe are being held here, safe and secure, ready to be used in the next time of crisis.
And now with Aleppo, we have seen that the Svalbard Global Seal Vault works as it should. When things go wrong, it is possible to go to the vault, retrieve the material, and then re-establish the collections.